This protocol provides a step-by-step procedure to sample the perivitelline membrane sublayers from avian eggs for further investigation of their physiological role in bird reproduction. The sampling of the perivitelline membrane has been optimized at each step to limit any structural and molecular damages. The protocol was further developed for in-depth proteomics.
Begin by sampling the perivitelline layer, or PL, from a freshly laid unfertilized egg. Break the egg, and use an egg separator to separate the yolk from the white. Remove the chalazi with small scissors, and roll the yolk over a filter paper to remove adherent albumin that appears as a transparent but visible structure. Immerse the yolk in a crystallizer containing 10 millimolar tris HCl at pH 8 that has been previously cooled to 4 degrees Celsius, and remove the PL area over the germinal disc within a 1 cm zone using blunt scissors. Rupture the PL with small scissors inside the buffer. Then, hold the two edges of the ruptured PL with forceps and peel it off the yolk. Rinse the PL several times in baths of 10 millimolar tris HCl until no trace of yolk is visible. Ensure that the PL is clean, white, and floating in the buffer. Process the PL for sublayer separation or proceed directly to biochemical analyses. To separate the OPL and IPL, spread the entire sample with the OPL facing up in a plastic petri dish filled with 10 millimolar tris HCl and 50